Welcome friend. Topic is your immune system. If you are facing the same problem that I used to face, that means I am unable to understand the basic of this immunology. So I've decided to read all those things for you and to try to help you to understand all those things in the basic format way. Okay, so let's begin from this immunity. So the first thing that I want to show you here is that innate immunity and adaptive immunity. This is the two types of immune system that found in a body. In case of vertebrates, you only see this adaptive immunity. I'm just saying the important points, so please try to remember all these things. Most of the interview you will ask this kind of question also. But in case of innate immunity, it's found in all the animals, but the adaptive is only in vertebrates only. So the pathogen using a small set of receptor, the pathogens here a broad range of pathogen are taken receptored by a very small or narrow range of set of receptor. But here in adaptive immunity, the recognition is using a vast array of receptor, and those are very specific, so very small range of pathogens being detected, but receptored. Then it is your rapid response, but in your adaptive immunity it is your slow response. Try to get all those things, please. Innate, so it's rapid in response, whereas adaptive is slower in response. Now the innate immunity is mainly found in barrier defense, internal defense, while there's some cell which is responsible for this barrier defense or because of that innate immunity. I don't know the answer. If you know, just comment below. The barrier defense and internal defense. In internal defense, you can see the innate immunity are like the phagocytic cells. That's the cell I want to say you. The phagocytic cells, natural killer cells, the antimicrobial protein, and inflammatory response. So these are related with your innate immunity. I have told the answer to you. So the phagocytic cells and natural killer cells, they are the cells which belong to the innate immunity. Okay, which were the phagocytic cells, the natural killer cells. Okay, in case of your barrier defense, the skin, the mucous membrane, secretion, all of this belongs to this innate immunity. It's nothing so. And adaptive immunity, there is a humoral and your cell mediated response. In humoral response, the antibodies defend against the infection in a body fluids. This is your humoral response. In case of cell mediated response, the cytotoxic cells defend against the infection in a body cell. We'll discuss coming slide. So lysosome is an enzyme that breaks down the bacterial cell wall for to protect this insect digestive system. So not like only insect, many other animals. This is the innate immunity of invertebrates I'm talking right now. Here is a nice picture is shown. Here is the toll receptor. First in is shown here is the pseudopodia surrounds the pathogen. Pathogen then capture with their phagocytic cells. I have told you in innate immunity there is your phagocytic cells and natural killer cells. So that's here I'm discussing the phagocytic cells which surround this with its pseudopodia, and by means of endocytosis it encloses pathogens inside the vacuoles, then fuse with the lysosome that here I'm discussing. So that lysosome merges with that vacuoles, and by fusing it digests with the help of its enzyme, and that's causes the release of this debris from the pathogens, and is released by this exocytosis method. Now I'm shown there's some also other technique. For example, for viral infection, the antisense RNA, or what we call RNA interference technique being used, that kind of techniques. So here what has happened, the host enzyme dissect to recognize this double-stranded RNA structure found during this replication cycle of an RNA of a virus. So this viral RNA being detected, and by this dissect to enzyme, it cuts this into fragments, and about that means it, and that fragment is about 21 nucleotide long. Let me mark this. We better see this. Okay, that's fine. In the second step, you can see the protein complex containing this host enzyme Argo binds to an RNA fragment. That fragments that will be clipped by this dicer of this viral RNA. A protein complex containing this host enzyme Argo binds to an RNA fragment, displacing one of these two strands. The RNA complex uses bound single-strand fragment as a guide matching to the complementary sequence in viral mRNA. So this RNA complex uses this bound single-stranded fragment as a guide to detect those viral RNA. So wherever there will be the viral mRNA will be there, it can be detected, and there it can clipped easily. And that's by this way it can block the synthesis of viral protein. So this is a mechanism of innate viral infection pathogenic activities. So the natural killer cell and phagocytic cells in internal defense barrier. So now it's time for adaptive immunity. Okay, one more thing left, sorry for that. It is a toll-like receptor. Each mammalian toll-like receptor recognizes a molecular pattern of characterizes a group of pathogens like lipopolysaccharide, flagellin, CpG isolate DNA, the double-stranded RNA also. So these are some things which will be receptor by this toll-like receptor, and then after is toll-like receptor protein trigger. This toll-like protein triggers this innate immune defense, including the production of cytokines and antimicrobial peptides. That goes and fight against all those things. So some cellular innate defense. This is the question I failed to answer. Okay, so here the toll-like receptor, mammalian recognizes protein similar to the toll-like receptor in insects. So neutrophil, the two main type of phagocytic cells in mammalian body, like a neutrophil and macrophage. Here neutrophils, which circulate in blood, are attracted by signals from the infected tissue and then engulf and destroy the infecting pathogens. The macrophage is a big eater. The neutrophil is circulated in the blood, attracted by signal from the infected tissue. There is a beautiful picture I'm going to show you in some little bit while, so just keep patience. So macrophage is a big eaters like the one who shows in figure. I will let me show. Okay, let's move on to this. Okay, dendritic cells, popular tissues, skin contact environment. So these are some cell about the natural killer cells. Cellular innate defense. This is cellular innate defense is the natural killer cell. Okay, this is circular throughout the body, detect the abnormal array of surface protein. Some of these viral infectable intracellular cells do not engulf the striking cell. Instead, they are released chemical that lead to cell death. So chemical released by these natural killer cells lead to the death of this infected cells. Okay, not much I want to tell you, but this is the thing. Okay, you can read out all those things. Interferon, complementary system. It is one important thing. This is a complementary system. This is how it's fine. Circulate surface. Okay, we'll just read it out. That's nothing. So this is picture of different lymphatic, primary, secondary lymph nodes, etc. Blood capillary, interstitial fluid, lymphatic vessels, etc. You better try to understand all those pictures written here. Okay, the main thing here I'm going to show you, yes, this picture. Let's begin the game. So inflammatory response, histamine. So this is picture very nicely being drawn here, and I want to tell you that there's an injury by this splinter. Pathogens, bacteria are also coming inside the tissue. So what exactly happened? The macrophage, which are present in the tissue, they in presence of in contact with those pathogens, they release some signaling molecules. Those signaling molecules with associated with that mast cell also some produce some signaling molecules. So here this macrophage secreting signaling molecules attract those neutrophil cell that is in the capillary to get attracted towards this wound. But here the mast cell producing the histamine, which cause this dilation of these vessels and cause the porous formation in capillary that helps to come out this neutrophil to the tissue, and there they by coming the phagocytes this all this pathogens, bacteria, whatever, and heal the tissue. So these neutrophils just digest the pathogen and the cell divides to heal the body. So this is your thing. So I have shown here somewhere, yes, here I have shown here the two main type of phagocytic cells in the mammalian body are the neutrophil and macrophage. They are the primary innate immune system, that is your the macrophage and the neutrophils. Here also written I'm focusing this two term because this is the most important type of term or question that being asked you during your interview. The phagocytic cells, the natural killer cells, neutrophils, etc. Okay, so this is the thing. Now come to this adaptive immunity. Receptor provide pathogen specific recognition. So this is okay, so it is the adaptive immunity. Okay, that's fine. So here is the structure of a mature B cell and mature T cell. So before going to this lymphocyte, this video getting longer, sorry for that. Okay, so lymphocyte, the lymphocyte has T cell and B cells, you know, and the T cell mature inside the bone marrow, where your B cell sorry for the B cell and T cell both produce from the bone marrow, but the T cell has to move to the thymus gland where it get mature. Okay, here is some antigen receptor on the B cell. You can see the antibody is on the surface of the membrane. And anything else, let me show you. I'm just showing you a picture because that's all that matter. This writing doesn't matter to you, not me. Okay, so here you can see there is some epitopes. Epitopes are the pathogenic recognition structure which is recognized both by these antibody paratopes. So B cell possesses antibody which can recognize those antigenic epitopes, and after it get recognize those epitopes, it releases antibody. So here it's antibody release. And the one more thing that has to remember is that a single pathogen has multiple epitopes, and but the B cell has only one type of antibody produced, and the two specific. So this is the thing. Different B cells have to secrete antibodies which is common to that pathogen having multiple type of epitope. That's the thing. Okay, and it is a T cell which has this T cell antigen receptor. So here it recepts this antigen fragment because T cell won't be able to directly bind with the pathogenic substances, but the B cell can do it. Okay, so how this cell bind works, I mean all these things work. Let me show you. Here you can see this is a MSC molecule. This is a major histocompatibility complex which shows this pathogenic, I mean peptide. Is this peptide? Yes, it is peptide, sorry. So this peptide fragment of this pathogen which is being ingested inside the host cell, and that MSC molecule is displayed on the fragment, and this can be recepted by this T cell antigen receptor. So this is your MSC shown in picture, and there is some variation that how this variable region of that antibody is produced by this whole recombination method. So some delete DNA, some transcription. So these are the processing here done. I'm not going to discuss all those things. Okay, so here's the important one, and once again, so there inside the body there are variation in B cells because those recombination of this what you call differential recombination of those variable and segment and functional gene which form these antibodies. So due to their combination, it produces various type of antibodies. So the B cells also there in our body is various in type, having multiple receptor. And as I have shown in the very first thing, the difference between inner and adaptive. The adaptive is very specific and very small range of receptor is there. I mean very large range of receptor for very small specific range of pure pathogens. So that's why it has multiple type of antibody receptor, I mean antigen receptor. So each has different type. This is a B cell having different type of paratopes antibody to bind with that particular type of epitope, and this is also another type. So only few are able to bind with that kind of pathogens. So once they're bound, very important, try to understand, once the selected B cell bound, those B cells which bound will be selected for cloning. Here is written, the selected B cell proliferate and forming a clone of identical cells bearing the receptor for that antigen. So then they differentiate into two types. One is the plasma cells which continues releasing those kind of antibodies. Another one is the memory cells which will store those differentiated cells for next secondary exposure. There must be a graph. I have, yes, this is a graph. You can see here, when exposure to that antigen A, it's an immediate action. The concentration of antibodies becomes so high because it's already been prepared during as a memory cell, so it won't take to it, so the time doesn't consume to make these clones because it's already being stored. So the number of antibody concentration is vigorously high as compared to the early stage of what we call the fast interaction or exposure to that antibodies, antigen. Okay, so now let's come to the helper T cell. Hopefully you getting understand what I'm showing. Picture very important. Writing a note. Okay, pathogen being inserted, being taken out, taken in inside the cell. And remember, there is a difference between antigen presenting cells and some other kind of cells. So the MSC class MSC2 molecule, class 2 major histone complex is only shown your antigen presenting cells, whereas your normal cell only produce the normal cell produce both class 1 and class 2 MSC molecule, but the antigen presenting cells only produce the MSC2 molecule, and that's the difference. You can say as a difference. Okay. Then another thing is when it's bound, it has to be bound with the CD4. So it is an accessory protein which helps to recognize those MSC molecule, and that's present on the T cell. That to be of your T helper cell. Okay, so CD4 class 2 MSC molecule, that is your antigen presenting cells. So this is your helper T cell. So what does the helper T cell do and how they do? Okay, so the cells, after binding, the cell which have taken this fragment will release some factors, and that factors helps to proliferate this cytokinin which produced from the T cells. Here's written: the binding of helper T cell promotes the secretion of cytokinin by the antigen presenting cells. The cytokinins along with the cytokines from the helper T cell itself activate the helper T cell and stimulate its proliferation. So the cytokinins are produced from the antigen presenting cells. That's a mistake by me. Okay, so the cytokinin is produced from those antigen presenting cells. Oh my god. Cytokines is produced from this cytokine produced from helper T cell and activate the helper T cell and stimulate its proliferation, and those what we call, oh my god, I am also forgetting, sorry. So now what happened, the T cell, the helper T cell is being proliferated, being activated by the cytokinins from that will be produced from that antigen presenting cells. So remember, okay, the binding of the helper T cell promotes the secretion of cytokinins by the antigen presenting cells, and the cytokinins along with cytokine from the helper T cell itself activate the helper T cell and stimulates its proliferation. Okay, now come to the next point, that here you can say the helper T cell has now to do a novel work. Now its function is to activate all the cells in a clone have receptor and this proliferation is also cloning type. So once this helper cell is ready to help this fighting it, so what happened, the helper T cells the cell secretes other cytokinins. So this cytokinin secreted from the helper cell. So now you can see the cell proliferation produce a clone of activated helper T cell. The cells in the clone have receptor for the same antigen fragment complex with the same antigen specificity. These cells secrete other cytokinins which helps to activate this B cell and cytotoxic T cell. Thanks, I just survived. Yes. Okay, so all cells in the clone have receptor for the same antigen fragment complex with the same antigen specificity. So these helper cells now secrete some cytokinins which goes and helps to activate those cytotoxic T cells and your B cells. The cytotoxic T cells is mediated the cell mediated immunity, and the B cells which produce the antibody is in your humoral immunity, as I have shown in this picture, that the cell mediated and humoral immunity. So B cell is for humoral immunity and cytotoxic T cell is for your cell mediated immunity. Okay, and this secretion of the other cytokinase which helps to activate this B cell T cell. So all the cells in the clone have receptor for the same antigen fragment complex with the same antigen specificity. The cell secretes other cytokines. The cell secretes other cytokines. Okay. So from here it also has cytokines being released. The T cell also released. Okay. Now the B cell and antibodies response to extracellular pathogens. So this is also another important diagram. So you can see here it is the helper T cells which are binding with that of your CD by means of your CD4 to that class MSC2 molecule. The cytokinin being produced helps those B cell, because the B cell is also an professional antigen presenting cells. So it only shows this MSC2 molecule on its surface by taking pathogen inside it. So this cytokinin that produced during helper T cell cause activation of the B cells. That's very important, because during this disease like HIV/AIDS, what happened? The helper T cell get suppressed, and that's where the immunity gets down. Even though the B cells are present, they are not able to get activated. So that's the problem. So this activated B cell now have two things to do. One, to make clones, and those clones have to separated. Some remain stored as your memory B cell, and some other will remain as your plasma cell, which secretes continuously the antibodies specific to that pathogenic epitopes. And those antibody, then what should they do? Because the antibody won't be able to destroy this pathogen of itself, so they helps in that way that the binding of antibody to antigen on the surface of bacteria promotes the phagocytosis by macrophages and neutrophils, because the antigen antibody binding causes the precipitation, and that the phagocytic cells like your neutrophil, the primary innate immunity causing substances cells, that is your neutrophil, macrophages, phagocytic cells, they what they do, they recognize those antibodies surrounding those bacterial pathogens, and they start phagocyting those pathogens. And here one more diagram is shown here. Those antibodies also secrete some membrane attacking complex, which forms some pore complex, which after activating this complement system, the membrane attack complex form pore in the cell membrane, allowing the water and ions to rush inside. The cell swells and lyses, and that's why the cell becomes burst. Here on the picture, I've told you that the class MSC1 molecule is being shown only in your normal cell type along with your MSC2 also, but the MSC2 is very rare. I mean, not rare, but little bit not produce in that much in that found in antigen presenting cells. So the class 1 molecule is only that found in your with little bit small amount of MSC2 also. Okay, so what happened? The class 1 MSC molecule will be recognized by the presenting antibody fragment, antigen fragment will be detected by the cytotoxic T cells. The cytotoxic T cells bind with that antigen presenting cells, that antigen receptor to that fragments with it help of this CD8 receptor to recognize those class 1 MSC molecule, and then it secretes the porphyrin and granzyme. Those granzyme and porphyrin cause the porous formation inside this in the cell membrane of your normal infected cells, and this is granzyme which will destroy those cells and initiate those apoptosis within the infected cells, and leading to this fragmentation of the nucleus, cytoplasm, and eventually the cell become death. So that's the importance of their cytotoxic T cells. Here is the basic picture. You can see I have discussed all those things, and this is the most important picture you should be go through, because it's contain all those things I have told in earlier. This is how the antigen presenting cells help to proliferate the helper T cell, to form memory T cell, to fight against the second antigen. Then your B cell, T cell, cytotoxic T cell activated, they cause the plasma cell, memory cell, bind and kills those cells in your humoral and cell immunity system. So that's it, man. Thank you very much. Nothing is all important. Thank you very much. Nothing. Thank you.